Hello dear learners, in this video we will continue with our topic mutations, and in our previous video we had covered the topic of spontaneous mutations. In this video we have to talk about the non-spontaneous mutations or the chemical and physical methods of inducing mutations in the genome. So first of all we have to discuss about the chemically induced mutations. Okay, so before we start with this topic, in my last video also, I made this very clear that what are wait, uh, yes, what are mutagens. This I covered in my last video also. So mutagens are those agents particularly which causes the mutation above the threshold level of normal mutations. Means there is a certain threshold level up to which the mutations can occur in our body, but if it is above that, then this is the induced mutation, and this is caused by certain agents which are causing that mutation, and hence these are called as mutagens. So we have to talk about the chemically induced mutations, which will be obviously done by the chemical mutagens. So there can be many chemical mutagens, but let's see that what can they do. First of all, either they can mimic the natural bases, means whatever are the nitrogenous bases which are located in the DNA, these chemical mutagens can mimic them, and they can have the certain modifications, and when they are inserted into the DNA, now they will cause the mutation. Or what they can do, they can even modify, they can do the process of modification. It means that they can modify the already existing nitrogenous bases. Also they can insert themselves in between the nitrogenous bases. Suppose two normal, like suppose adenine and guanine was there, and this chemical mutagen inserted itself in between them, and it was pretending as if it were also a normal nitrogenous base. Then also it can do the oxidative damage, and oxidative damage is the most harmful one. If the oxidative damage occurs, then the cell has no other way, it has to only perform the apoptosis, and this is the internal pathway of the apoptosis, wherein the cell will then have its programmed death. So these are the functions what the chemical mutagens can do. Now there are various types of chemical mutagens, one of them being the base analogues. In the normal nitrogenous bases, if there is a certain modification because of which they turn into a different kind of nitrogenous base, or if they turn into a, suppose say, a different analog of that nitrogenous base, that time we say that it is the base analog. The best example here can be 5-bromouracil. We know the structure of uracil is like this. It is already a pyrimidine, so it has this kind of structure here. Here is a double bond, and here we have double bond O and double bond O. This is a normal uracil. Now if by chance what happens is here the sorry, not methyl group, bromine group, if this bromine group is added here, that time we say that it has now become the 5-bromouracil, and this double bond will be removed after that. So this compound which is so formed, it is known as 5-bromouracil, and this 5-bromouracil can insert itself into the normal DNA sequence, and it can also cause the mutations. Now there is a very special feature of this 5-bromouracil. What happens is that this particular 5-bromouracil can do the base pairing with adenine. Now suppose this is my structure of adenine. Suppose this is my structure of here we have NH2, it will have its NH here. Okay, so this is a general and the cyclic structure of a purine, and then here we have this particular NH2 group is added, so we can say that it is adenine. For the clarification, I can also draw these double bonds here. Now what happens is that this particular hydrogen gets the hydrogen bonding with this nitrogen, and here this oxygen so present here will do the hydrogen bonding with these hydrogens. So here what is happening is that the 5, wait, I'll change the color. 5-bromouracil, whatever is present here, it is 5-bromouracil. It is doing a double bonding with the adenine here. So it is mimicking as if it were also a natural base, and in that condition it can create much of the problems. So one of the example is this. The another example of certain base analog is 2-aminopurine. So 2-aminopurine is also one such example. So this can also have the similar kind of structure here. 2-aminopurine also can have the chemical modifications, and it can also become a base analog of purines. So these are the first kind of chemical mutagens, that is the base analogs. Now we should move about to the second kind of chemical mutagens which can be present, and those are the alkylating agents. Now the alkylating agents are those whose main function is to add the R group to the particular DNA sequences, or in short to the particular nitrogenous bases. One of the examples here is ethyl methyl sulfonate. Now this ethyl methyl sulfonate will add the ethyl group, that is CH2CH3 group. This group will be added to the guanine. So this ethyl methyl sulfonate acts as the alkylating agent. So it is a chemical mutagen. It will add this ethyl group to the already existing guanine, and that time when it does it, so it will cause a replication error also. So what it forms from this ethyl methyl guanine, what is the product form? Sorry, from this ethyl guanine, what is the product form? So the product name is O6 ethyl guanine. This is what the product is formed. Now this O6 ethyl guanine, we know that normal guanine has to have the triple bonding with cytosine, but this O6 ethyl guanine does the double bonding with thymine. So it creates a mutation. The another such example can be the mustard gas. The mustard gas is also one of the alkylating agents, and this can also, if present in higher concentrations, can cause the mutations. So this is another kind of chemical mutagens, that is the alkylating agents. Then there are certain chemicals, direct chemicals, which can cause deamination or which can cause the hydroxylation also. So let us discuss about them. One of them we will talk about first, the deamination agents. Those agents which can cause this deamination. Now we know that the structure of cytosine has to be like this way. Right, this is the structure of cytosine. Now what happens is, if this is treated in the presence of HNO2, now please remember this, that this HNO2 here will act as a deamination or deaminating agent, and this will remove this particular NH2. So here this NH2 will be removed, and now what happens is, instead of this NH2 here, the oxygen gets the double bonding with this carbon. So here we get this type of structure. And now you can easily guess what is the structure. This structure is that of uracil. So from the cytosine, in the presence of deaminating agent, we got uracil. Now this uracil will then do the bonding with the adenine, and that bonding I have already shown in the previous slide. So there can be certain agents which can cause the deamination. Then there are certain agents which can even cause the hydroxylation. So yes, there are certain agents which can cause hydroxylation also. For example, if I talk about hydroxylamine, this is the best example for this. So what is there in hydroxylamine? The formula for hydroxylamine is NH2OH. This NH2OH will do the hydroxylation, and particularly this will do the hydroxylation of cytosine only. So if the hydroxylation has been done, so what happens? The final product of cytosine in the presence of NH2OH becomes hydroxylaminocytosine. Hydroxylaminocytosine. So here there are certain important points which you need to learn. One of them being that the deamination agent is HNO2. This HNO2 will act upon cytosine. So now you see, you can learn it with this part that both deamination and then this hydroxylation, both of them perform by or which are performed on the cytosine. So in the deamination process, what is occurring? This NH2 group which is present here, it has been removed here, and instead of that the O group is added. And in the hydroxylation which is carried out by hydroxylamine, that is NH2OH, what happens is again on the cytosine it is acting, and NH2OH is acting, so it will form the hydroxylamine cytosine. So these are other two types of chemical mutagens. Then after this, so this was third and fourth. Now after this there can be another cases also. One of the mutagens can be acting as oxidative agents, and the oxidative agents will directly damage the DNA. So they can also act as the chemical mutagens. And now what happens in the, like for example, if we take the example of guanine in the presence of oxidizing agents will be converting itself into 8-oxy 7,4 dihydroxy guanine. At least you have to learn one example from each of the chemical mutagens, and that will also suffice your answer. Now this will do a mispairing with adenine. Now if you remember from previous slide, I was talking about the alkylating agents where I talked about ethyl methyl sulfonate. There also it was modifying the guanine and it was forming O6 ethyl guanine, and that guanine was base pairing with thymine, and this time this guanine is base pairing with, or basically mispairing with adenine, but traditionally it has to pair with cytosine. The last type of chemical mutagens which can be present is the intercalating agents. The intercalating agents are those which will insert themselves in between the base pairs. Suppose these are the base pairs. Suppose here I have the base pairs of a DNA. Now what happens is, in these base pairs, one of the agent coming from outside gets inserted here. This agent was present outside and this is inserting itself here. So it is performing the process of intercalation and insertion, and this agent is known as intercalating agent. Now there are many examples to it, one of them being proflavin. Now proflavin acts as the intercalating agent, the another one being the acridine orange. All the dyes which we use to basically stain up the genetic material, all those can also act as the intercalating agent. Similarly, what we do and what we use in gel electrophoresis, that is ethidium bromide, that is also the calating agent, and one of them being dioxin. So four of them you can learn. One is dioxin, one is proflavin, one is acridine orange, and one of them being the ethidium bromide. So they will insert themselves in between the nitrogenous bases of the DNA sequence, and this will cause the distortion of DNA. All the structure of DNA will be distorted because of the addition of this intercalating agents. So these are your certain chemical mutagens which can cause the mutations. Now we should discuss about certain physical mutagens which can also cause the induced mutations. So let's discuss about physical mutagens. See, there are only basically there are two kinds of physical mutagens, and there also we have to just discuss about radiations. So one of them being the ionizing radiations. So I'll compare it here only. Wait. One is ionizing radiations. and in the comparative format, yes, the another one being the non-ionizing radiations. So there can be two, one of them being the ionizing radiations and the another of them of being the non-ionizing radiations. Now if I talk about the ionizing radiations, the examples of the ionizing radiations include one of them being X-rays, then another one of them being gamma rays, and even your cosmic rays. All these kind of rays are your ionizing radiations. Now what is the meaning of ionization here? Ionization means they are just trying to remove the electrons from the atom. They are just trying to ionize the atoms. So all of the atoms which are present in DNA, what do? Is they dislodge the electrons from these atoms, and that's how we can say that they are causing the ionization. Also they can do the wrong base pairing because now the electrons have been dislodged, so the original structure of bases have been already destroyed, and in that condition the proper hydrogen bonding will also be not taking place. Also they can break the phosphodiester bonds. Phospho wait, yes, they can break the phosphodiester bonds also, and also they can alter the base structures because they are dislodging the electrons from the atoms of these bases. So if they are dislodging the electrons, then it is very difficult to repair. That's why even if we talk about the Chernobyl accident which has occurred there, none of the human population lives there. Why? Because all those rays which are still present there, including these gamma rays and other ionizing radiations, they will cause the dislodging of the electrons from the atoms of the nitrogenous bases, and that time that is very difficult to repair, and it will cause permanent mutations and even death in certain cases if the effect is very severe. Then another being including the non-ionizing radiations. The best example here is ultraviolet rays. Ultraviolet rays are considered as non-ionizing radiations. They have comparatively less amount of energy as compared to the ionizing radiations. Now one of their target where they act is on the pyrimidine. So what do is they cause the pyrimidine dimerization. So this pyrimidine dimerization is done by these non-ionizing radiations. Now we know that pyrimidines can be cytosine and thymine when I talk about DNA. So either, like suppose if I am talking about, this is my DNA sequence, here is a cytosine present and here is a thymine present. So it can cause a dimerization between them also. So either cytosine dimerization, thymine dimerization, cytosine thymine dimerization, any of them can take place because of these UV rays. Generally these dimerizations, because these are only the hydrogen bonds, so they can be destroyed and they can be repaired. So many of the proof reading enzymes, they will repair this, but some of them may escape from this repair mechanism and then they can cause the mutations. That's why we say that the continuous exposure in the UV rays can also be very damaging. So this is there. At last we can say that there are these many detection method these many chemical mutagens which can cause the mutations in the normal genome sequences, and also these physical mutagens can also cause the mutations. Now these are the parts of the non-spontaneous mutations. Now we have to talk about one interesting method, and this interesting method is known as the CLB. This L is small. CLB method. This CLB method is done in Drosophila, and this is basically used to detect the mutation in the Drosophila. It is just a small experiment whose main aim was to detect the recessive lethal mutation. What kind of mutation? Recessive means in the presence of dominant gene it won't be able to express, and then it is lethal, that in case if it is expressing, that time it will lead to death, and then it is recessive lethal mutation. So this is your CLB method of Drosophila. Now what happens here? So First of all, we should decode this meaning of CLB. So CLB basically stands for, C stands for crossover suppressor. Crossover suppressor. Now basically what happens are, there are certain chromosomes in the Drosophila which are called as X chromosomes. Now a normal female will have the XX and the male will have XY. So there are certain genes which are present on this X chromosome, which are the lethal genes, recessive lethal genes. In this experiment we are mainly focused upon the X chromosome. So this X chromosome have to have the three characteristics. One is C, one is L, and one is B. C characteristic tells about the crossover suppressor. These suppressors have to be present on the X chromosome so that they inhibit the crossovers. And this X chromosome should be inherited as it is. See, basically what we are doing is, we have to detect the recessive lethal mutation, and we are designing this experiment artificially. So we want that whatever lethal gene that we are taking into consideration should not be exchanged and moving into unknown cell during the crossover. So what do we need to do is, we need to prevent the crossing over of this X chromosome so that the lethal gene which is present on the X chromosome is not able to move out of that X chromosome. So that's why we are introducing one of the suppressors here, which are called as crossover suppressors. So this should be the property in the X chromosome present if we have to do the CLB method. Then the L stands for the lethal gene. This lethal gene has to be present on the X chromosome. If it is not present, then we cannot say that there is any mutation there. And lastly, we talk about B. B is the bar-eyed gene. If you remember, in my last to last video I was talking about the bar gene mutation, wherein we were talking about the duplications, where double bar was also present, or bar duplication was present. So those bar genes which are present on the X chromosome, they are also included here in the consideration of this experiment. So see, if the lethal gene is present, the bar gene has to also be present. Now we cannot determine whether the lethal gene is present in the Drosophila, in the progeny of the Drosophila or not. But yes, definitely we can see it by their eyes. If the eyes are bar shaped, that means these Drosophila which have the bar shaped eyes, they will also contain this lethal gene. So in this X chromosome, there has to be this crossover suppressor, then there has to be a bar gene and associated with the lethal gene. So the lethal gene is inherited into the offspring, the bar gene will also be inherited into that offspring. So that we can have the check easily, we can check easily by just analyzing the eyes of those progenies. If the progenies have the bar shaped eyes, it definitely means that they also have these lethal genes in them. Now, how do we start up with this experiment? In this experiment, we take first of all the male Drosophila, and now this male Drosophila is treated with a certain kind of mutagen. This male Drosophila will be treated with a certain kind of mutagen such that its X chromosome may contain that mutagen. Now it can be present or it cannot be present. We are not sure. So one of the things which we take in this experiment or in this method is that we have to take a male Drosophila which is introduced to that mutagen. Second important thing is we have to take a carrier female of that mutagen. Now we know that in the females there are two X chromosomes, so at least one of the X chromosome must have that lethal gene. And since this lethal gene is recessive, it won't be able to express itself in this carrier female. Now what do we do is we are trying to cross these two. Now here we are not sure whether that particular thing is present or not. Okay, now what happens is that when we are crossing it, there will be the males produced, and all those males which are produced here, most of them which have bar eyes, they will die because they have only one X chromosome, and this X chromosome is inherited from the mother. So if they have inherited this X chromosome, so they will have the bar eye, and in that condition they will die. So this can clearly detect that the mutagen has been expressing itself, and it is from that recessive X chromosome only. This recessive lethal gene has expressed itself in the male progeny and it has come from that X chromosome. Now what happens is, in the second experiment, now we take a normal male. And also remember that whatever females which are produced here, these females, if they die, if some of the females start to die, that time we can say that the parent male was also having that mutagen. And if the females do not die, that time we can say that the parent male was not having that particular mutagen and only the female was having the mutagen. So that time they can die or they cannot die, it's up to them. Then in the next experiment we take the normal males and then we cross it with these females. Suppose these females, then if they're not dying, then they definitely have to be the carriers because the mother was a carrier. Now when they are crossed, what happens is, in the another ones, means in the next progeny whatever we are getting it, that is the F2 progeny, if that F2 progeny wait, consists of all the females which are alive and consist of the males where half of them die and half of them survive, that time we say that this particular female was also a carrier. Means what we are trying to prove from this experiment is this particular male Drosophila which was introduced to that mutagen, whether this mutagen is capable enough to induce mutation in it or not. And that we can easily determine from this very first experiment only. From here only we could have easily determined that how this mutagen would have been acting. But the second experiment performed is just again a confirmatory experiment only. So this is a simple CLB method which is used to detect the recessive lethal mutations in the Drosophila. With this, our mutation unit gets over.